Hi, my name is Ryan McEwan. I'm professor and Shoeline Endowed Chair at the University of Dayton in the Department of Biology. Here I'm going to present to you Seeing the Forest and the Stream, Amur Honeysuckle Effects on Headwater Streams and Forests. This is going to be a synopsis of more than a decade of research in my lab that's focused on this species. Invasive species cause human or economic harm. By definition, they are an important threat to biodiversity. This is an image of silver carp leaping in a waterway, just to give you a visual for the high density and sort of alarming character that can be present for some invasive species. This particular fish is a very serious issue in some areas in the American Midwest right now. The species I'm going to focus on is Linicera mackii, Amur honeysuckle. It's a shrub originally from Asia. It was introduced to the United States in 1896, and there was a Soil Conservation Service breeding program that was involved with this species. The idea was to develop this Asian shrub for soil stabilization and wildlife purposes, and ultimately this escaped from those plantings and now has become one of the, if not the most important invasive shrub in the Midwest of the United States. So I want to give a shout out to Dr. Rachel McNish, who did a lot of the work I'm going to present to you today. She is presently an assistant professor at the California State University in Bakersfield as an aquatic ecologist. Also, Dr. Kevin Custer, who is presently at Pace Analytical in Englewood, Ohio. He is a postdoc on this project. And the work that I'm going to be presenting really was absolutely not possible without the leadership of these two individuals. Also, I want to start off by thanking the National Science Foundation, who funded a large portion of the work you're about to see. I'm going to make some main points in this talk. The first is that Amur honeysuckle fundamentally changes the structure of forests along stream banks. What we have with a honeysuckle invaded stream is fundamentally different than what you have if native species only are present. Amur honeysuckle contributes large quantities of materials to streams that are toxic to some macroinvertebrates. Macroinvertebrates are organisms that live in streams, are very important to stream function. Amur honeysuckle contributes quantities that are toxic to at least some of those species. Amur honeysuckle invasion changes the aquatic community in fundamental ways, is my third point. The last point is that restoration efforts, even at smaller scales, seem to create positive response in stream organisms. These are the four main points of the talk, which I will now go through one after another and present to you some evidence that supports each of these points. First, Amur honeysuckle fundamentally changes the structure of forests along stream banks. Part of this is visually obvious, at least here in Dayton, Ohio, where we see honeysuckle invasion across forest streams that oftentimes looks like this, where there's literally a cave of branches over the top if you're standing in the stream and looking up. This is Eric Borth, who worked in my lab for many years. Here's some images from spring and summer. You can see the Amur honeysuckle trailing across the top of these streams, creating a virtually impenetrable leaf layer. In some cases, the leaves are literally trailing down into the water. This creates a structural situation that is not present with other native species. Here's an image from winter to give you an idea of what a heavily invaded stream can look like. Again, basically all of the wood you see above that stream in this image is from Amur honeysuckle. This experiment was a complicated one. I can't share with you all of the details from it. I can only say that we measured many different things: herbaceous seedlings, tree seedlings, saplings, woody debris, leaf material, and many other things. And this was the structure of our plots. And we had Amur honeysuckle on some of these plots, and it was a design that was like this. We have reference streams where Amur honeysuckle is absent. You can see no honeysuckle there. We have the moderately invaded streams where we have Amur honeysuckle present along the stream, but not in great quantities. And then we have heavy streams that have that sort of cave effect, which I showed you in the previous image. Particularly for some of the data I'm going to present, we had 5 streams that ranged in invasion intensity from reference where honeysuckle is absent. You can see on the left there, moderate to heavy, and the heavy stream you can see is just incredibly overrun with Amur honeysuckle. Across the top of this design are native trees. So the native tree canopy is overarching the stream and overarching the Amur honeysuckle. This species invades as an understory shrub. So we have to just put the native tree canopy there before you can understand the design. So putting that native overstory canopy across the top of these streams, we have reference streams, moderate streams, and heavily invaded stream. And again, this work was led by Dr. Kevin Custer, who was a postdoctoral associate. Also, this particular part of the project was led by Aaron Roykamp, who was a master's student many years ago in my research laboratory. Aaron was specifically interested in the idea that woody debris, and you can see woody debris on the ground there in front of her, she's taking some measurements, that woody debris could be influenced by the presence of this structure of the Amur honeysuckle overarching the stream. And she found evidence of this actually in her master's thesis research. In this particular graph on the x-axis is Linister macii basal area. That's how much honeysuckle is present. And you can see the green points there where you've got Amur honeysuckle increasing in intensity this way. And on this axis, the y-axis is coarse woody debris volume. And there is a relationship here that's a negative relationship. As you increase Linister macii in the across the y-axis, there is less woody debris in the streams. This is in-stream woody debris. Increasing honeysuckle in the forest was associated with decreasing woody material in the streams. We had observed this also in the field. It looks like this. It's hard to get a good image of this. I've tried many times, but literally we have woody debris suspended in the Amur honeysuckle branch structure suspended over the stream. Here's another image, and you can see here, this is relatively large tree. This is maybe 12-inch diameter tree that was captured and stuck there in the Amur honeysuckle branches. Woody debris plays a critical role in the biology of streams, and our data suggests this may be influenced by the physical structure of the honeysuckle cave effects over the stream. This is an interesting finding. It would be a great thing to follow up on, but it does suggest that the fundamental biology, the infrastructure of the stream has been influenced by the presence of this invasive shrub. That's point number one. Number two, Amur honeysuckle contributes large quantities of materials to streams that are toxic to some macroinvertebrates. So here's another image of streams where honeysuckle is present. This is not quite the cave density, but we do have Amur honeysuckle on the banks of this stream. And all you can see here in this, all this material in this stream bed, that's flowers from Amur honeysuckle. In the spring, there's a great contribution of flowers into stream systems and here in Dayton, Ohio. And in the fall, of course, especially late in the season, there's a foliar deposition that's also very considerable. And the phenology, the leaf time for Amur honeysuckle is extended when compared to native species. And so you can see Amur honeysuckle foliage deposited into streams into December. It can hold its leaves into December. So you've got this deposition of material coming into the stream. Also, an interesting attribute of Amur honeysuckle is it has incredible quantities of fruit. And here you can see a underwater image of some of those fruit in a headwater stream. We don't know. There is some potential that the seed can be dispersed this way through the water system. That's called hydrochory. There's definitely nutrient effects because those are nutrient-rich little seeds. And there's also potential for toxicity associated with the fruit. So in this part of our research, we use multiple assessment techniques, including microcosm experiments that were both in streams and in a growth chamber in a lab. So this is an example of our, here you can see my cursor. This is an example of our in-stream microcosm. There would be an organism that we've placed into this structure. And in here you can see some fruit. And then there's sediments. Here you can see what that looks like, the rack looks like while it's in the stream. So you have a flow-through system. We also had systems that were in a growth chamber in our laboratory. So that would be a ex situ. What we found with this experiment is that as you increase the amount of fruit, that is the fruit biomass of Linister macei, the mortality of some macroinvertebrate organisms increases. So more fruit, more mortality, both in field and in laboratory settings. The take-home point, as the biomass of honeysuckle materials increases in the microcosms, mortality also increased. Here's another paper that follows the same kind of experimental design. This was a microcosm experiment that was only done in the lab. What we found here again is that as you increase the solution, you increase the Amur honeysuckle material in these microcosms, that survivorship decreases. So again, more evidence of toxicity created in aquatic organisms by increasing material from Amur honeysuckle, increasing the concentration of material. The take-home point is that as the biomass of honeysuckle materials increases in microcosms, mortality of these organisms also increases. This is evidence of toxicity in both a field and also a laboratory setting. The third point, so that's point number 2. The third point is that Amur honeysuckle invasion changes the aquatic community in fundamental ways. So back to our reference moderate and heavy stream design. We went to these streams and we assessed the macroinvertebrate community that is in situ. We also, by the way, did leaf pack experiments where you literally put leaf packs containing honeysuckle and native trees and then mixtures of honeysuckle and native trees in streams. I won't present those findings. It's a bit too much to be contained here. But this particular data set I'm going to share with you is from literally going to the stream and sampling aquatic macroinvertebrates across a range of different intensities of Amur honeysuckle invasion. We brought those samples back, sorted them, and this is just one bit of data from this research, where we have in the heavier sites where there's more honeysuckle invasion, the percent EPT taxa is lower and statistically significantly lower than in all other sites. The moderate sites, and this is kind of a staircase control. This is a reference site with honeysuckle absent, the moderates, and then the heavy sites. This is obviously some difference between these two reference sites. However, the general pattern suggests that as you increase the amount of Amur honeysuckle in the riparian zone, going from none present to some present to very heavy invasion, that you're changing the percent EPT taxa. EPT taxa is a way of measuring the sensitivity. More sensitive organisms are absent in the heavy site. This leads to this overall, we've put all of the work we've done together on macroinvertebrates. It leads to this overall concept of what's happening in these headwater streams. We have changes to structure and terrestrial inputs on the bank as Amur honeysuckle invades. This alters the energy and nutrients and also the habitat substrates of the stream. This changes the basal resources that are available for aquatic organisms, whether it is coarse-woody debris creating habitat structure, or whether it's toxicity, the toxic contents in some of the materials. And as the basal resource for aquatic organisms is altered, that creates a bottom-up effect on the aquatic food webs, because macroinvertebrates, of course, are part of a broader food web. And we have evidence to suggest that this shifts the composition of the community in fundamental ways. The last point is that restoration efforts, even at smaller scales, seem to create a positive response in stream organisms. In this particular experiment, what we did is a honeysuckle removal before and after kind of experiment. Here is Rachel, Dr. Rachel McNish again, who is the person who led this experiment. Here we have water flowing this way. This is a headwater stream. And these little puffs here are Amur honeysuckle. Here's the native forest canopy again. We have to put that on top like this. So here's a stream that has the stream, the honeysuckle, and the native forest. And our design was to remove Amur honeysuckle over a portion of this riffle, so we would, or this stream, so we would have controls and we would have removal. And then we were sampling in the riffles for macroinvertebrate composition. And again, we have to return the native forest over top of it. So we have a removal reach and we have a honeysuckle reach that looks like this. One thing to add is that the sites we were working in, the honeysuckle was incredibly dense. It was like this. You almost feel like you could lay on top of it. It's very, very thick branches. And what we found in this study is that when we removed Amur honeysuckle, there was a response in the macroinvertebrate community where the macroinvertebrate density increased, and we found a unique, and this is a ordination diagram over here. It's a little hard to see, but these ellipses are not overlapping. We have a unique macroinvertebrate community in the removal reach versus a honeysuckle reach, and we also have an increase in overall macroinvertebrate density. These data indicate that removing honeysuckle along a single stream can have a positive effect, even if Amur honeysuckle is still present in the stream farther upstream. So for example, if you're a manager of a nature preserve and your property ends, but the stream is flowing through it, if above the preserve there's honeysuckle present, you can still remove honeysuckle in your preserve and have an effect that's measurable on macroinvertebrates. I do want to give you some caveats on that bit. First is that we were working in streams where the riparian zone was relatively uniform and was forested. I want to be really clear about this, that other factors like parking lots or agricultural fields are more important to stream biology than Amur honeysuckle. Okay, so if you were to look at streams across a range of different conditions, and some are right beside the city street, some are right beside a parking lot, some have concrete instead of substrate, Amur honeysuckle effects are not going to be the same as an ag field or a parking lot. So what we're talking about here is comparing forested sites with and without honeysuckle present. Also, we were working in streams with very heavy honeysuckle invasion. There is literally a cave, not literally, but seemingly a cave of honeysuckle over the top of many of these streams in Dayton, Ohio. And we've done a lot of stream surveying in the region, and there's a lot of honeysuckle. I would say it's fair to say that streams in a lot of Ohio are characterized by Amur honeysuckle invasion in forest. And restoration activities must consider interception surfaces. This is a really important point. It's probably too detailed for this talk, but if you're going to remove Amur honeysuckle, if this talk has led you to think, you know what I'm going to do? I'm going to take Amur honeysuckle out of my nature preserve. You need to think about, what are we going to do? What are you going to do to intercept the rainfall? Because the issue is that directly under Amur honeysuckle is probably just mud, because the honeysuckle kills or suppresses native species. That's a whole other topic. But Forest ecologists have long since established that Amur honeysuckle has negative effects on herbaceous plants, for example. So if you go in a densely invaded honeysuckle site and your feet are probably muddy, it's like there's just mud. So if you remove the Amur honeysuckle, now you've just got mud on the banks, and you're going to have a lot of siltation potential. So I just want to encourage you, if you hear this talk and you think I want to do restoration or removal, to think about what's the interception surface and plan that ahead of time. So establishing some sort of native plants, you can consult with your local botanist. Something like impatiens, which are touch-me-not, it's a native riparian plant that grows very fast, or think about sedges. There's lots of things to think about as an herbaceous layer. And then I would also urge you to think about native shrubs. Okay, spice bush, pawpaw, witch hazel. There's a wonderful shrub called Viburnum prudifolium, which is rusty blackhaw. There's lots of interesting native shrubs. Those shrubs should be the target of conservation efforts in general anyway. So I would really urge you, if you're going to do removal, to think about interception surfaces, establishing native plants, even maybe before you start the removal. I don't want anyone to take out Amur honeysuckle and then have a terrible siltation event because you haven't thought of this. These are my main points. Amur honeysuckle changes the structure of forests along stream banks. It contributes large quantities of materials to streams that are toxic to some macroinvertebrates, not all for sure, but some. That invasion changes the aquatic community in fundamental ways, and that restoration efforts, even at smaller scales, seem to create a positive response in stream organisms. With that, I want to thank many undergraduate researchers who worked in my laboratory over many years, and who have gone on to success in various fields, not just ecology, also ophthalmology and other places. It's wonderful to work with all these students who've helped us advance this research. And if you have any questions, please reach out to me at ryan.mcEwan@udayton.edu. Thanks very much, and visit our lab at mcewanlab.org.